Okay, you're on.
All right. Can I acquire some applause? Decentralizing science. Development of a cure for cervical cancer. Yes. All right, salutations. My name is Zachary Swady, and as evinced by my title, my 20% project was developing an original treatment for cancer with biotechnology. And in doing this project, I realized something imperative concerning the biological community, the scientific community that I intend to share with you today. I was inspired to do this project on account of the fact that biotech that provides a feasible treatment for cervical cancer has not actually been developed yet. I realized also, I also was inspired by the fact that cervical cancer is such a severe disease. In this presentation, I first shall discuss the biotechnology that I designed. I designed it base pair by base pair, 20,000 base pairs. It was hellish, but and I shall subsequently notify you of the issue that I located in the scientific community. So section 1, a nascent biotechnological approach to selective apoptosis induction in cervical cancer. Within this section, I shall elaborate upon my treatment proposal, my biotechnology that I designed. An overview, right? So I separated the section into two subsections. The first, which is why I attempted to cure cervical cancer, and the second, which is how I attempted to cure cervical cancer. Well, so first section. Allow me to commence with the more obvious, more comprehensible of the two first, by providing you with a characterization of the effects, the mechanisms, and the failings of the current treatments for cervical cancer. So allow me to provide you with a deceptively odd fact. According to the CDC, in 2014, 12,578 women were diagnosed with cervical cancer, and 4,115 women died from cervical cancer in the United States alone. Perhaps you find this curious when considering that HBV causes nearly all instances of cervical cancer, and that safe sex has become a more common practice in most regions throughout the years. However, when we consider uncultivated regions, rural regions, undeveloped countries as well, 80% of cervical cancer cases occur in undeveloped countries. It becomes less surprising, and that, when analyzed with the current cervical cancer survival rates, allows us to comprehend the severity of the disease. So the survival rates for pre-cancer, which is on the slide 0, 1A and 1B are essentially approximately 80 to 93%. Yet in the most severe manifestations of cervical cancer, such as 4A and 4B, survival rates can decrease to as little as 15 to 16%. This decrease is such a diminutive survival rates. They're being precipitated by the failings of current cervical cancer treatments, and also their inaccessibility. Such treatments include hysterectomies, which frequently cause infertility, the total infertility, and yet possess suboptimal success rates. So is it worth doing? Probably, cervical cancer, but still. And also radiation treatments, which also possess severe side effects that can actually result in cancer again. I mean, my grandfather, he actually underwent radiation therapy for cervical cancer, or not for cervical cancer, for pancreatic cancer, and I think it was for pancreatic cancer. But he developed a recurrence in somewhere in his body. However, with radiation therapy, again, it is 50% success rates for even the 1A, 1B, and 2B, the most, the least severe of the cases. But wait. Why am I doing this? Why am I attempting to vindicate curing cancer when you are likely aware of its detriments already? Actually, can I get a show of hands? If any of you have been are familiar with someone that has been touched by cervical cancer, raise your hand, or any cancer actually. Is that a hand? All right, precisely. That is nearly all of you. So you are aware already. Why should I be explaining this? I mean, however, if you are not, I do possess a video that I prepared to elucidate the detriments. So consider for a moment this cyclist to be a patient, and regard this door as cervical cancer. There we go. There's supposed to be sound. Anyway, he hurls himself through the door, okay? It impacts him, quite literally. So indeed, cervical cancer is severely detrimental to both patients and families, which is evinced by the video, of course, with the severe impact. Anyways, just to summarize, current treatments are unsuitable when considering the implications of recently developed biotechnology. That is a quote by me. We require a cure, comrades. So my proposal, utilizing biotechnology that can cure cervical cancer by selectively inducing apoptosis in cancer cells. So, of course, if any of us did the HBV project, Ragu Komalapati, we know that that HBV causes cervical cancer by utilizing its E6 and E7 oncoproteins to degrade the P53 tumor suppressors and cause tumor development, which these anthropomorphic Patrick tumor men represent. So I think actually that the presence of the E6 and E7 can be utilized to our advantage, however, in curing it. That is why I selected to utilize synthetic biological circuits. They are essentially these plasmids, these pieces of circular DNA that are programmed to recognize the presence of a molecule in a cell and in response produce a specific output, that the operations of which are quite similar, very much similar to that of an electronic circuit. So I thought that we could you take E6 or E7 as our input, recognize the presence of E6 or E7 in the cell, which of course would be indicative of cancer, of cervical cancer, as they cause cervical cancer. And in response to the presence of those, output pro-apoptotic proteins, pro-apoptotic molecules and such. But this diagram, I thought that was quite cool. So in order to cause apoptosis, though, it is somewhat of a complex process, since in cancer cells it is often prevented by certain mechanisms, such as multiple nuclei and certain proteins that inhibit apoptosis. So in order to do this, then, we must produce both apoptosis inducers, such as the proteins BAX and BAD and BID, all of these, BIM, PUMA. So essentially what these do, with PUMA, for instance, it inhibits other anti-apoptotic proteins by binding to them and such. And with BAX and BAD, they are effector proteins, also with caspase 3, that actually cause the apoptosis. And the second component, inhibiting the anti-apoptotic proteins and genes, such as BCL2 and BCL XL and BCLW and BCLB and MCL1 that inhibit the apoptosis inducers. So in order to do that, in order to inhibit the genes, we must utilize a concept that I developed, an intracellular CRISPR system. with CRISPR, is essentially a complex of Cas9 and gRNA. The Cas9 is guided by the guide RNA to a specific location on the gene and removes the DNA from the gene. And it can be utilized to disrupt genes. So for one of the outputs in my circuit, I chose to utilize a CRISPR system that would, hence intracellular CRISPR system, that would be able to edit these genes with gRNAs. So it actually took, it was actually a bit difficult to design as it required 5 separate gRNAs and the Cas9, which was 4,000 bases, so that contributed to my 20,000 base sequence. And also, yes, it was highly specific. So designing the intracellular CRISPR system. So I utilized something that's referred to as multiplex expression with the 5 separate sgRNAs that guided the Cas9 to specific locations on the genes, on the BCL2 genes, the ones that I mentioned. So what it would do is the Cas9, again, it would be able to disrupt the gene by cutting, by recognizing the locations and sort of removing segments of the DNA from the genes, which, again, this was a bit odd. It was a bit difficult to produce, and so now I shall discuss synthesizing the entire circuit, which involved, so actually, as opposed to utilizing the E6, E7 as my input, I revised it to utilize the A3B, or this lengthy name promoter that is actually activated by E6, E7, which provided, allowed me to utilize the, to actually sense the presence as opposed to just it. So the A3B allowed me to sense the presence of the E6, E7 protein since it was activated by them. With my outputs, I utilized BACs, all those pro-apoptotic and anti-apoptotic proteins, or no, pro-apoptotic and the anti-apoptotic molecule inhibiting proteins, and also the intracellular CRISPR system. So again, to produce it, I utilized a combinatorial design program that essentially I programmed the circuit. I programmed a truth table for the circuit, is right here, and it analyzed it, and in response calculated the various layouts of the circuit, calculated and selected which one would be the least toxic by analyzing the inputs and outputs in combination with these bacterial gates, these biological gates that essentially controlled the circuit. This is the entire diagram of the circuit. The lines are bacterial OR gates, so if the A3B is present, it allows that to occur. It allows all of these to occur. However, if it is not present, it does not allow any of them to occur. Sort of circuit. This is the complete sequence. Actually, this is it. 1/3 of the sequence, 20,000 characters. It's disturbing. I cannot even examine it without it becoming sort of ill. I attempted to download the file, and it's such a substantial file. In order to deliver the circuit, though, I sort of thought of something sort of similar to an antivirus. Antivirus in a computer software. So essentially, what I thought of doing was utilizing cloning the plasmid into lentivirus, which would be utilized to infect all of the cells in the cervix, and thus recognize the cancer with maximal efficiency. Yes, so, and that can actually be optimized for other cancers as well, since the A3B promoter is activated by other oncoproteins and such. There's that smiley face. So the section summary, I designed a nascent biotechnological technique to essentially treat cervical cancer, and however, it became quite difficult to produce, to actually realize the technique, which is what I cover in this section. So why it is nearly impossible for a high school student to develop such technology. So producing the circuit, my initial attempts were contacting certain nonprofits, certain firms, such as AdGene. They specialize in CRISPR systems. So I contacted them. They referred me to this company, Thermo Fisher Scientific, which I pursued. Also referred me to Atom and Origin, some other companies. So my experience with them, I discussed services with them. They were willing to provide me with to allow me to utilize services despite to allow me to utilize their services despite not being a member of any institution, being just an independent independent actor, just an honors biology student. So what I did was I emailed them the sequence for a quote, so they could provide me with an estimated price, and they returned a quote of $6,000 US. And so that infuriated. That is what I became. I rose and didn't know. Interactions with other companies. I contacted Atom. I telephoned them. They never returned an email to me. And with Origin, I don't even know. I emailed them thrice. They have not responded. I finally accepted the inevitability that I would be unable to produce the circuit in the month that was allotted for circuit production with the approaching deadline of the 20% project. And so I delayed production until a science fair that I'm doing in March of my sophomore year. Possibly by then, I could develop the circuit, but I must pray for a research laboratory position, which also it would require me to be in a lab that is capable of doing synthesis, and those require experience in order to be in, which obviously as a high school student I possess absolutely none of, and again, they frequently do not even accept high school students. So even with Worcester Academy, even if I attempt to develop it at Worcester Academy, the Worcester Academy cannot culture cancers, cannot culture mammalian cells, if I'm correct, and the HeLa cells that are required to perform experiments would be $500 or more, and they can also not fund me $6,000 US to purchase a circuit. So,
who did you ask?
Cannot call what?
Who did you ask?
You. So I must pray.
Oh my God.
So this is what I am calling for in response to this. I think that perhaps in order to cure cancer, we must provide everyone with an opportunity to cure cancer, including high schoolers, and that can be done by providing them with a discount, by not charging $6,000 to produce a DNA sequence. So that is why I request that you invite me to your laboratory.
Oh my God.
Thank you.